So my name is Winnie, and I am currently a third-year biology major. And today I'll be talking about cancer pharmacology, or understanding how cancer is regulated by drugs. And so the specific cancer that I'll be talking about today is B-cell precursor acute lymphoblastic leukemia. And I had no idea what it was when I was researching this either. And so here's a little schematic. I'm sure you've heard of blood cells. You've heard of red blood cells that take on the oxygen. You've heard of platelets that clog up. And then you've heard of white blood cells, which are part of your immune response. And so B cells are a type of immune blood cell. And this is the most common type of childhood cancer with a 90% 5-year survival rate. And so that sounds very high to you, but because it is such a common cancer, drug-resistant leukemia is actually the 4th most common type of childhood cancer. And so our research focused on understanding the differences between those leukemias that are sensitive to drugs and those that are resistant. And currently the treatment includes cortisol hormones. And so for us to be able to understand what the differences are, we need to understand how cortisol works. And here is taking you back to high school biology, talking about the central dogma. And so here you have your DNA, which is your cell's genetic information. So that's where you code all of your genetic information, and then it gets transcribed into something called mRNA, which is sort of like a messenger that brings the genetic code outside of the nucleus into the cell, where you can make protein. And protein are the functional units of a cell, which allow for your everyday functions to happen. And so cortisol, how it works is cortisol comes into the cell and then binds to these specific proteins that then go into the nucleus and bind on your DNA. And so once it binds to your DNA, it has some different types of functions, one of which can be transcription regulation, and it can turn on specific genes or it can turn off specific genes. And the gene that we're specifically looking at, let's just call it gene X, it's being turned on when cortisol binds. And so I arbitrarily just said your cortisol somewhere binds on the DNA. And so you might be asking, well, where does it bind? And that's a very good question. And so let's imagine this is your strand of DNA and that is where your gene is. Well, would it bind right here, close to it, or would it bind like somewhere over here and farther away? How would we know? Well, if you imagine your DNA in this 2-dimensional space, you might say, oh, well, this is much closer, so it makes sense to bind here, right? But in reality, your DNA is much more like a big bowl of spaghetti. It's very flexible, and it wraps around itself, makes knots and whatever. And so in 2-dimensional space, yes, this might be closer than somewhere over here, let's say, but in 3-dimensional space, this might just wrap around over here and be even closer. And so that is essentially how your DNA looks like. And so this is, you can say in 3-dimensional space, this might be quite far, but if it wraps around like this, then it might actually be very close in space. And so we're trying to figure out where to look. And so our methods, first of all, is figure out where all this cortisol is binding. And so we've mapped the genome, found out where cortisol binds, and then what? So there's all these cortisol binding sites right here, 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 and here. How do you know that this specific cortisol binding site is actually regulating this specific gene and not this one, or maybe a combination of them? Or how do you know that it's not regulating some other gene that's closer by, or some other gene that this one might just regulate that one instead? So what we've done is we've induced mutations to these specific sites, and then we've looked at the transcription levels of our gene of interest. And so you can imagine that because the cortisol comes in and binds to DNA and turns our gene on, what happens when you mutate this site right here, then that means that transcription would be halted, or there'll be less transcription. And so what we've done is we've just mutated a bunch of sites and compared the mRNA content after transcription. And here we have some data, and over here on the right, you see this is our gene of interest. And in certain sites, you do see that there is a loss of regulation, which means that the mutations have caused there to be less mRNA produced. And in certain sites, the mRNA content, it's still there, which means that that site might not be regulating our gene of interest. And so we found that was quite interesting, and so we decided to put these sites together and mutate a combination of sites, because in biology it's never just one thing leads to another. It's always a combination of multiple things happening at the same time, and so we decided to mutate multiple sites, and this is what we see here. And you can see that if we've mutated multiple sites, loss of regulation, some of them are still there. However, those with mRNA that if you mutated sites and the sites were actually not regulating, you see actually an increase in the mRNA content, which means that if you've mutated sites but they're not interesting, then the combination of all these sites together actually causes an increase, which is quite interesting to us, but this is all preliminary data, and we don't really quite know what's going on there, and so that is for future research. And so future research, what we want to do here is we want to make sure that we're repeating our research and talking about the importance of reproducibility, because what's happening one day in the lab might be different from another day. What's happening in our lab might be different from another lab. And so reproducibility is super important. And then next is to find out, okay, we've established that we have all these sites that cortisol may be binding to. What's next? Bringing it back to understanding sensitive versus resistance, we might look at these sites and say, oh, what's the difference between sensitive and resistant cells at these specific sites? There might be different proteins, might be different downstream regulation effects that are happening that we don't know of, but once we've established these sites, then we figure out what's the difference between the two different types of leukemias, and then maybe we can figure out some drug therapies that may be able to help. And so one last thing that I want to say is that although we're looking super deep into the molecular level of biology, understanding transcription and genetic regulation has many implications far beyond pharmacology and far beyond cancer biology, because if you think about it, a lot of diseases is inherently just a misregulation of something. And so that provides insight into different ways in which we can look at different diseases, and also it can provide a really good platform for understanding social determinants of health on a macro scale, because basically the environment that you're living in, the type of jobs that you do, what you're exposed to, that is all comes together and affects the molecules inside your body. And so, yeah, that's all I have for today. Thank you.
Great talk, first of all. Just curious, when you were doing your mutations, was it in cells or in animals? Or?
Yeah, so we did mutations in cell culture, so it's just cells in a laboratory. Yeah.
All right. So why use cortisol as a marker instead of any other?
Right now, the drug treatment is the cortisol hormone, and how cortisol works is it turns on specific genes in B cells that cause cell death.
So is cortisol, in a way, a drug treatment for those B cells?
So yeah, right now the drug treatment is cortisol for these 90% survival rates. However, for the drug resistant, the cortisol is not working, and so we're trying to figure out what the difference is and how that works.
I wanted to ask, so, cortisol has a range of functions, including, it can stop bone formation and stuff. So when you're giving children cortisol hormones, what is the procedure to do it so they're not permanently affected by it? Do you know what the concentrations are?
I don't know specifically what the concentrations are for treatment, but I do know that there are a lot of side effects for using cortisol hormone treatment, because cortisol hormones not only regulate cell death of your B cells and your immune cells, which cause the anti-inflammatory response, but also fat proliferation or bone degeneration and all that. And so that's what a lot of the doctors take care of on that side of the treatment, but we're really trying to understand the difference between the treatments in drug-resistant and sensitive leukemia. And so that's all inside like a cell laboratory. And so if you're asking me the concentration I use on my cells, then I might be able to tell you, but in a real human being, that's a different question.
So what concentrations do you use in your cells? Like what's the thing for resistance? Like what's the concentration for resistance? How much are they resistant to?
So we're just using cell culture of B cells right now. We're not actually using patient samples. So, yeah.